Hi, everybody. I'm Megan Malden, your Saline County Extension Agent with the University of Arkansas Cooperative Extension Service, and I am here today with Dr. Heidi Ward, who is the University of Arkansas Extension Veterinarian. And Heidi has agreed to do a 5-part vet science series with us this month, which we're really excited about. And this 5-part series, as many of you already know, is covering animal senses. So she's covering a different sense every week, the sense of sight, smell, hearing, taste, and touch. And this week's video, today's volume, we're going to be covering how animals hear. And I know you guys are excited. I certainly am. And I'm just going to get out of the way here, and I'm going to let Heidi start talking and do her thing.
Well, hi guys, we're on week 3 and talking about hearing. We've talked about the sense of smell and the sense of sight and the different cells that are involved in that process and how it communicates with the brain and how we react to it, not just as humans, but then comparing everything from humans out to animals. And we'll do the same thing here. We'll talk about just how we hear first and then compare other animals to us and what the differences are. But first we need to talk about sound. Actually, first let's talk about this cat right here, because I have my subtitle, Is That a Can Opener? So you see the cat's look on its face. They always seem to know. They could be anywhere in the house and they hear a can opener, they know, is that tuna? What is that? And you will find out that cats have a very good sense of hearing for a variety of reasons, but a lot of it has to do with their ears. But before we talk about that, let's talk about just sound in general. Sound is measured in hertz. So it's about sound frequency, and sounds come in waves. And the lower the sound, the deeper the sound, the longer the wave. And you see how they actually spread apart a little bit. They're wider. And then the higher pitch, higher frequency, so something that's really, really high and squeaky, is going to be in that really fast, short waves. And then, of course, and that's just the distance between the wave, top of a wave to a top of a wave. Now, the taller these guys are, because these could still be far away but really tall, that's going to be volume. So we have volume and we also have the pitch. So when we're talking about sound and everything we're talking about comparing the animals today, we will be talking about hertz, or the frequency, or the pitch. Okay, so let's talk about us. What is our normal range of hearing? Our normal range of hearing is 20 hertz to 20,000 hertz. We can also call that 20 kilohertz. So 20 hertz to 20 kilohertz is our normal range of hearing. Anything below that 20 hertz, we cannot hear. There are some animals that can hear in that low range. And then anything above 20,000 kilohertz, we cannot hear. That's above our range. So those really high-pitched noises, like mice, the high squeaky things that we can't hear. Let's just compare right off the bat. Dogs can hear from 67 hertz to 45,000. So they can hear things that are much higher pitched than us. Cats, way higher pitched than us. Mice, higher pitched than us. Bats, really higher pitched than us. They can hear things all the way up to 200,000 hertz. That's the highest range. Birds are pretty much in our range, but there's one particular bird that we'll talk about at the end that can hear some really low frequencies. That is pretty cool, and they're around us all the time. Whales use very low frequency to communicate in the oceans. And elephants also use very low frequencies. And a lot of that is because these waves, these low frequency waves, can travel a further distance. And that's why these animals that are far away from each other can use that particular frequency to communicate. And we can't hear that. So let's talk about our ear first. When sound waves come in, so that's little sound waves coming into our ear, the first thing it encounters is the pinna. The pinna is the outside of our ear, the floppy part, and its whole purpose is to direct those waves into the ear canal. So we're directing the waves into the ear canal. Our ear canal is just a straight shot, just a straight line. And that's why we're always told not to put Q-tips in our ears, because we could poke some very sensitive things. And this is the sensitive thing we're talking about, and that's the tympanic membrane, also called the eardrum. The eardrum is connected to some auditory ossicles. Auditory means hearing. Ossicles, or really anything when we're talking anatomy, if it starts with os, it means bone. So they're little tiny auditory bones. And then those communicate with the vestibular apparatus, which is for balance, and then the cochlea, which is what processes all of those sound waves to the brain to determine pitch and volume and all those things. Let's take each one of these one by one so we can understand them a little better. Oh, and of course, then all the information from the cochlea and the vestibular apparatus go to the brain through nerves. And it's the vestibular cochlear cranial nerve is the main nerve that goes to the brain to transmit. But there are other nerves for different areas. So let's look just at these auditory ossicles. So remember, those are the little bones of the brain. These little blue circles are the sound waves, and the first thing it hits is the tympanic membrane, or the eardrum. That's the red that you see there. The first ossicle is the hammer. The medical term is the malleus. So the hammer does exactly what it sounds like. It is a hammer that hits the second bone, which is called the anvil. So they are the incus. The medical term is the incus. Now, they get these names because of what they do. The hammer hits the anvil, and some of you younger ones may not know what an anvil actually is. An anvil is a device that was used back in the ancient times, and even now, by blacksmiths to hammer out metal. And so here's a picture or animation of a guy with a hammer hammering a sword. Making a sword on the anvil. So that big metal block that he's hammering on is the anvil. The stirrup isn't really the function that it's named for. The medical term is the stapes, but we call it the stirrup too. When you look at these actual bones, and here's how tiny the human ossicles are on the fingertip, the stapes looks like a stirrup. And when we think of a stirrup, those are stirrups that we use when we're riding horses that you put your boot in. So that's where it got its name, the stirrup. So really not because of its function, but just because what it looks like. And then of course the cochlea. So the waves hit the tympanic membrane, the hammer hits the anvil moves and moves the stirrup, which communicates with the cochlea through the oval window. That's the special term, but just through a particular area where it transmits further. So now let's go deeper into the cochlea. And you'll notice that the cochlea is shaped like a snail. Well, guess what? Cochlea actually means snail in Greek and Latin. So cochlea is named after a snail because it looks like a snail. We name things for what we know. If we take a little cross section, so what this is we cut through a little section of that cochlea, and now we're looking straight at it. This is what it looks like. But as the wave goes through, the messages go through, and this wraps all the way around, it gets smaller and smaller and smaller and smaller in diameter. So this right here will get smaller and smaller as it gets deeper into that spiral. There's a reason for that, and we will talk about it. It contains 3 chambers, and what you see on this diagram, there's the vestibular chamber, there's the cochlear chamber, and then there is a tympanic chamber. We're only going to focus right now on this one right here, which is the middle one that you see going all the way up through here that's in orange. And that's where the information from sound actually gets transmitted. You'll notice on this diagram, it talks about perilymph and endolymph. So these different chambers have perilymph. Lymph just means liquid, and that's body liquids. And any type of liquid that's outside of a cell is considered lymph. Perilymph, all things are basically perilymph. The only thing that makes endolymph different, especially in this chamber, is that it's really high in potassium. Why is it high in potassium? It's because that potassium is used to transmit those electrical signals through the special cells inside the cochlea. And these cells are called, dun, 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 hair cells. Isn't that crazy? Hair cells. I always think the ear and hair, just, you never want to think about hair in the ear, but hair cells, they have their reason, their name, because of the projections they have that look like little pieces of hair. And we'll show you a close-up of that too. But when we go close up onto that, really all the transmission happens at those hair cells in a special organ that's at the bottom of that inner cochlear duct, and that's called the organ of Corti or Corti. And that is where the actual hair cells sit and where the movement of that endolymph from, remember, the last thing that hits is the movement of that stirrup hitting that oval window, pushes that fluid along and makes waves in that fluid, and then those waves move these hair cells. Okay, now let's talk about how that relates to pitch. Okay, this is how it relates to pitch. So the higher frequencies, so the really high-pitched sounds get transmitted early on in the cochlea. So those are the hair cells that are sensitive to those high pitches. That long waves, the longer the waves, the lower the pitch, goes really, really deep down into the very end. So the very end, that smaller diameter, has all those hair cells that are sensitive to those lower frequency waves. So high pitched at the beginning, low pitched down at the bottom. Okay. Now, when we talk about listening to music too loud or things being too loud and we get hearing damage, most of that damage, by the way, is to these high frequency hair cells. And we do have a lot of low frequency hair cells that still work. You may notice if you hear ambulances these days, they do have a special siren on them now that has a really weird, funky, low buzz to it. That's because a lot of people that have become deaf, especially the elderly, cannot hear that higher frequency anymore, but they could still hear some of those lower frequencies. So there's reasons behind a lot of that, right? Now let's talk about the vestibular apparatus. This one's kind of fun. So it's really cool. It's really cool looking. It has 3 semicircular canals, and they're all in different directions, different orientations, and they're filled, again, with perilymph. They're filled with lymph, and also lined with hair cells that actually can feel, just if you're standing still, you know, they can tell just from gravity and the movement of that fluid what position you're in. And it's mainly trying to figure out the position of your head and how it's moving in relation to gravity, but only when you're still. So I know I'm up and down, but if I'm laying down, it's these semicircular canals that's getting that information. We'll look a little closer on how that works. There's also 2 pockets that are on the inside that are actually the otoliths. There's 2 different pockets. One's the utricle and the saccule. You don't have to go deep into what those differences of those are. Just know that they are in charge of the dynamic movement or dynamic equilibrium. So the semicircular canals is if you're just sitting still and it's using gravity to find out what position your head is in. But the utricle and the saccule is actually figuring out where you are when you're moving. Okay, what direction's going when you're moving. And that has to do with some special little crystals and how it interacts. We're going to blow this up because I know this picture is a little hard to see, but we'll blow it up so you can see exactly what it's doing. But with either way, either in the semicircular canals or in the otoliths, it's using hair cells are the ones that are sending that information. And then what do they do with that information? Well, they take the information, take it to nerves that go to the cerebellum, which is a special part of the brain that's right at the back of the brain that is only responsible for detecting and coordinating movement and balance. Cerebellum, very important. We're going to talk more about that. So let's first blow up what you see in static movement. So this is what happens in the semicircular canals. And what you see at the bottom, we have the hair cells, and they have these little projections, which we call cilia, that actually float around in this special little compartment, fluid-filled compartment called a cupula. Okay, so this is one big giant fluid-filled compartment, and then there's other fluid outside of this too. And then when the sound, when you move, or when you're just sitting there, gravity is conducting the flow of the fluid through these semicircular canals, and it pushes these cupulas in one position or another, and that's how you detect through these 3 different orientations exactly what position your head is in. And then there's also supporting cells. The support cells are just that. They support the hair cells. They don't conduct any information to the brain. And then in the cochlea, well, not in the cochlea, in the otoliths, this one's different because you still have the hair cells and the support cells, but now you have this kind of gelatin matrix where these crystals sit on. And then when you move, these crystals slide down and push down and stimulate these hair cells and cause those cells to be excited and send information through the vestibular tract to the cerebellum. So 2 different ways, static equilibrium and dynamic equilibrium. Now let's talk about what happens when it gets to the brain. All of the information from the pitch sound through the cochlea goes to the auditory cortex, which is this little tiny area on the side of your brain, which is called the temporal lobe. That is where the auditory cortex is. And then the vestibular cochlear cranial nerve is the main nerve that sends information from the vestibular apparatus in the cochlea. Some of it goes to the auditory cortex, and then some of it goes to the cerebellum. So cochlea goes to the auditory cortex, and vestibular signals go to the cerebellum. So all of that information gets projected onto the brain. We're going to talk about pretty quick, another part of the brain that gets stimulated through hearing for animals that use echolocation. But first I want to talk about the cat ear. The cat ear is somewhat similar to human ears. It does have just one long canal, but it's not a straight shot. It just is kind of like a very, very slight bend to it. They have a pinna, an ear canal. All the inner ear is the exact same how it works as a human does. But cats have 32 muscles in each ear. Cats have muscles that they can use their ears independently, and they use that in order to hear. And I'm going to show you an example of a cat that hears a noise, and he gets one ear and he essentially closes it down. So he knows if I think I hear it here, if I close it down, if I hear it again, and it seems louder, then I know it's coming from that direction. But if I close my ear and it gets quieter, then I can move my ears to see if I can find it a little more. So here is a cat doing that. So he hears something, closes that ear. Oh, it's over there. Now he knows. So that's pretty cool. So cats have that advantage, and you know most of the animals. We talked last week that the rhinoceros has some pretty cool ears on the top, and he can use them independently as well. Rhinoceros can do that. Horses can do that. Dogs, not so much. And dogs have all kinds of different ears. Yes, they can move them, but they don't move them like cats and some of the other animals can. Another cool thing about cats is their cerebellum. You notice that cats have amazing sense of balance. If you look at our brains, our cerebellum is maybe, takes up a good portion of our brain down at the back, at the bottom, but nowhere near in comparison of how much of the brain is dedicated to cerebellum in a cat. So a cat has a much bigger cerebellum in comparison to the size of its brain than ours. So they can use a lot more information in their cerebellum to coordinate movements with their eyes and with their bodies. A lot of information goes to their cerebellum, which gives them amazing balance. We don't have that. The dog ear is different than ours because it is shaped differently. They can hear higher pitches than us. They can't hear some of the lower pitches that we can, by the way. But I wanted to just point out that they have a very severe L shape. In fact, we divide up their ear canal into a vertical canal and a horizontal canal. And a lot of times as veterinarians, we see them for ear infections because this elbow right here, they could get a collection of a lot of wax and hair that blocks the air from getting into this horizontal canal, and they could get inner ear infections from that. Well, actually, it's going to be outer ear infection still. We don't consider it inner ear until we get over here. And it's usually yeast and bacteria that can collect there and cause an infection. So the story of the blue-eyed cat, just wanted you to be aware that blue-eyed cats, 65 to 85% of white blue-eyed cats are deaf, and that's just from mutations that due to their coloring that has to do with the cochlea. So the alleles that are responsible for giving them blue eyes and white hair also happen to be responsible for the development of the cochlea and those hair cells, and they have mutations. They just don't work. And then cats that have just one blue eye, if they are white, now it's only white cats, if they are white and they have one blue eye, 40% of them with one blue eye are deaf. So, blue eyes and white cats do not mix. Just want to let you know that. And then the really interesting thing is if they have just one blue eye, that they are deaf in the ear on the same side as that blue eye. So if we see that one blue eye, we want to know if they're having hearing issues, we can just test the hearing in that ear. It's done through electrodes. It's not a scary test, but turns out the ones that are just deaf in one ear do just fine. We also have an issue with genetics in certain dog breeds, and the main one is Dalmatians. And there is a common misperception that all Dalmatians are deaf. They are not. 8% are born deaf. There are multiple genes involved, so it's really hard for breeders to find out by genetic testing if they'll be deaf or not. But what we do know through science is that the mutations are involved with the melanocytes. And if you know what melanin means pigment. So the dark spots you see on the dog are caused by melanocytes, and we do know that melanocytes are for some reason also important for hair cells and the development of hair cells. So they have some issues with their mutations in their melanocytes is what actually makes them Dalmatians, right? Gives them that spotted appearance. But guess what? It also messes with the hair cells. So 30% are deaf in one or both ears. We do know that about 5% of Dalmatians are deaf in both ears. And they're not born deaf. They can actually hear once they're born, but the hair cells die a few weeks to months after birth. So it's really weird how that works with the Dalmatians. And then what about echolocation? Echolocation is using sound waves. So they actually emit sound by either squeaking. It's usually a high-pitched sound. A lot of times we can't hear it. And then the sound bounces back, reflects off of something and bounces back, and that's how they can see it. So it's based on the reflection of sound. We talked about bats don't use their eyesight as much. They use their echolocation to see things. Blind humans use echolocation too. So the walking stick, the blind stick that they're using, they're tapping and using echolocation through the reflection of sound to see. So you can see through hearing, and there's a reason why, and it's pretty cool. It has to do with what part of the brain those messages go to. So these animals and people, by the way, who use echolocation have been able to send messages from our ears to a special part of the brain called the superior colliculus, which is in the same center where vision goes. So they send actual information from their ears to the same areas of the brain that process vision. So they are seeing by sound, which is cool. I think that's awesome. So last slide, which animals hear the best? Okay, so we know that bats and dolphins are the best at hearing because of their echolocation, because they can actually hear sounds and they have that added echolocation. Barn owls are one of the best because of the shape of their face. They have that weird facial disc. And in case you're wondering, where the heck's his ears? Well, birds don't have pinnas. They just have big old holes in the side of their head. So if you move their feathers back, you'll just see a big old hole. But that goes into an ear canal, and then they have the inner ear apparatus just like we have. Elephants and pigeons have that really low frequency. Pigeons, there are some pigeons that can hear the lowest, like 0.5 hertz, super, super low. And we use them they can detect earthquakes, low waves and sound from earthquakes. So pigeons, pay attention to pigeons because they can hear stuff coming when we can't. Cats, they hear well because of their ear movement, so that includes horses as well. Rats and mice can hear that really high frequency. So now you know about animals, how we hear, how animals hear, why animals can hear better than us, whether it's different sounds, whether it's the movement of their ears, or whether they use echolocation. And if any of you guys have questions, I would be happy to answer them right now. Megan, do we have any questions?
Yeah, right now we don't have any questions in the chat. I'll give it just a minute in case anybody wants to ask anything here at the end. I did have a quick question here, Heidi. So what video are you planning for next week?
Well, next week, let's see. We've done smell, we've done sight, we've done hearing. How about next week we do taste, the sense of taste?
All right.
That one's really interesting because that ties into what foods animals like to eat. And it ties into our last one, which is touch. So whenever we think about taste and we think about animals tasting things and what they'll eat, not only is it is it taste, but it's also texture, which is touch, and temperature, which is touch. So we'll kind of add that in. So next week will be taste, and then we'll lead that into the very last one, which will be touch.
All right. Well, thank you so much, Heidi. I don't see any questions, so that must mean that you were just super thorough. You answered all of our questions already. Thank you so much for talking to us, and we look forward to seeing you next week to talk about the sense of taste.
Yay! See you next week.
Thanks, guys.